In this video, we'll be talking about DNA replication as it has to do with prokaryotes. So we'll see that initially our helical DNA is going to separate apart, all right, and that's going to start our origin of replication, all right. And now if you imagine that this, instead of happening here where I'm just showing it, it actually happened over here, right? It's split apart. We have our origin of replication. That turns into our replication fork. This helicase enzyme is going to be splitting the double-stranded DNA into two single strands. All right, and those single strands are going to be held apart and stopped from bonding back together again by these single-stranded binding proteins that are going to be on each strand. And then there's a lot of tension and stress caused by the formation of supercoiling from the separation. So this enzyme gyrase is going to basically just alleviate that stress. All right, and then our next enzyme helicase is going to be here, and it's going to be putting on primers onto this strand of the DNA, right? And those primers are going to allow for RNA polymerase to come in and actually bind to and start replicating DNA. So this is where things will get a little different. We have one primer, we'll see one primer initially up here. This is going to be our leading strand. So it initially just has one primer, our DNA polymerase 3, which is coupled with a coenzyme that allows it to attach and move down the DNA strand. So that DNA polymerase and that coenzyme make what's called a holoenzyme. All right, it bonds, and it is going to be catalyzing phosphodiester bondage between the 3' end of the old molecule and the 5' end of new nucleotides onto the strand. So what that's going to look like is we start with our 5' end, and then this is our 3' end, so it's going to move 5' to 3' as it's replicating the DNA, and it's just continuously attaching the 3' end of old molecules to the 5' end of the new nucleotides. All right, and that strand is continuously replicated. That's why it's called the leading strand. Down here on our lagging strand, it's a little different. So this is going to be our 5' end, and this is the 3' end. So we're going to see our RNA polymerase 3 comes in, attaches here. It's going to move down, all right, replicates all of those nucleotides, synthesizes all that new DNA, and then a different complex, so now RNA polymerase 1, comes in, it removes our RNA primer and replaces it with DNA, and that leads to the formation of Okazaki fragments, right? So once there's an Okazaki fragment, this new enzyme ligase comes in, and it'll go here. It will basically replace the missing sugar bond between the nucleotides of the fragments and bond them together, so then they're a continuous piece of DNA. And so if we didn't have that action happening, then you would see that the primers wouldn't be removed from our DNA, and replication wouldn't be able to happen.